Good afternoon, everyone. I am here to present an overview of the features of bacteria, and provide multiple choice questions based on the material. I am confident this presentation will be both informative and entertaining. Let's start. Bacteria are the most abundant organisms on Earth, comprising nearly half of all life forms. Bacterial cells are comparatively small, from 0.2 to 10 micrometers in length. As prokaryotes, they do not have a nucleus, but do have their own set of genetic material, known as the bacterial genome, that is stored in the nucleoid. Bacterial cells are also surrounded by a cell wall of peptidoglycan to offer structural support. Gram-positive bacteria have an added outer membrane called the cell capsule, made of polysaccharides and proteins for extra protection. Inside bacterial cells are structures such as ribosomes for protein translation and flagella for movement. Despite their basic arrangement, bacterial cells can metabolize a wide range of substrates and execute the same fundamental tasks found in more complex organisms. Electron microscopy has provided us with an incredible insight into the structure of bacterial cells. Through this method, it has become apparent that bacteria are vastly more complex than initially thought. Differentiation between species can be found through the shapes, sizes, and external features of cells. Further to this, beneath the outer shell are multiple distinctive structures, such as the nucleus, the cytoplasm, the plasma membrane, and even flagella or pili. This inherent complexity gives bacteria the means to thrive in a variety of habitats. Bacteria have flagella, which are small and whip-like filaments that are essential for movement. This type of motility gives bacteria an advantage, as it allows them to move in reaction to different environmental conditions. This adaptive sensing behavior aids bacteria in finding food sources and staying alive. Bacteria vary greatly in shape and size, and four of the main types of bacterial cell arrangements include Lophotrichus, Paratrichus, Amphitrichus and Monotrichus. Lophotrichus bacteria feature one or two rows of flagella, while Paratrichus bacteria have flagella that are spread around the circumference of the cell. Amphitrichus bacteria have flagella located at both of the cell's poles, and Monotrichus bacteria have only one polar flagellum. Understand the different types of bacteria is essential for understanding microbiology, which is why the slide presented is titled, Bacteria. It shows four types of bacteria with each possessing unique characteristics. Monotrichous bacteria have a single polar flagellum, while Amphitrichous bacteria have a single flagellum at each end of the cell. Lophotrichous bacteria have two or more flagella at one pole of the cell, and finally, Paratrichous bacteria have flagella distributed over the entire cell. The shapes and sizes of bacteria are largely dependent on the type of flagella they possess. Flagella, a type of appendage that allows movement, come in two distinct forms when present in bacterial cells: Paratrichous and Lophotrichous. Paratrichous bacteria have flagella distributed all over the cell surface, while Lophotrichous bacteria have flagella located at either ends of the cell. Both forms of flagella give bacterial cells the ability to move through fluid environments and attach to surfaces. Plasmids are found in bacteria and are small, circular DNA molecules containing typically fewer genes than the larger bacterial genophore. Plasmids can confer extra abilities to bacteria, like antibiotic resistance, producing toxins and enzymes. Plasmids are extra-chromosomal circular DNA molecules that differ from a bacterium's chromosomal DNA. They have the ability to replicate themselves independently, storing genetic information including antibiotic resistance and metabolic pathways. Plasmids even allow bacteria to transfer them from one bacterial cell to another, granting bacteria a major evolutionary benefit. For this reason, plasmids are increasingly used within genetic engineering research and biotechnology. Bacteria come in a variety of shapes and sizes, and can be divided into different categories depending on the structure of their flagella. Monotrichous bacteria possess a single flagellum situated at one end. Paratrichous bacteria have flagella spread out over the entire surface of the cell. Lophotrichous bacteria have flagella at both ends, and amphitrichous bacteria have one flagellum located at each end. Knowing these structures is fundamental for studying the wealth of diversity found in the bacterial kingdom. The question regarding the small circular double-stranded DNA is answered by plasmid. Plasmids are able to replicate outside conditions of the chromosomal DNA and are capable of exchanging genetic material between different cells. Moreover, plasmids are useful in genetic engineering as they are able to introduce traits to host cells. Bacteria synthesize molecules, such as proteins, lipids, or nucleic acids, necessary for their survival and reproduction from nutrients in their environment. The relationship between bacteria and nutrition is complex, as they require certain compounds from the environment to fuel metabolism and growth. Essential elements like carbon, nitrogen, phosphorus, and sulfur must be obtained from surrounding sources such as soil, rivers, and air in forms that can be utilized such as organic compounds and inorganic ions. Bacteria are also able to use energy from light and chemical compounds for metabolic reactions. A thorough comprehension of the ways bacteria obtain and use essential nutrients is essential to understanding their development and growth. Bacteria can be classified into four main groups depending on their sources of carbon and energy. These groups are photoautotrophs, chemoautotrophs, photoheterotrophs, and chemoheterotrophs. Understanding the nutrition of these different bacteria is essential for research. Let's take a closer look at each group. Photoautotrophs use light as a source of energy and carbon dioxide as a source of carbon. On the other hand, chemoautotrophs use inorganic compounds as sources of both energy and carbon. Photoheterotrophs use light as the source of energy while organic compounds serve as the source of carbon. Finally, chemoheterotrophs use organic compounds as the source of both energy and carbon. Bacteria like Chromatium and Chlorobium have the unique ability to generate chemical energy from light. These photoautotrophs acquire carbon from atmospheric carbon dioxide and are of grave importance for the global carbon cycle due to their transformative role in converting carbon dioxide into organic compounds, usable by other organisms. Chemoautotrophs possess remarkable resilience and can thrive in harsh conditions. They can obtain energy and carbon from various sources such as oxidizing inorganic compounds and carbon dioxide, and thus do not require solar energy or organic nutrients for sustenance. This original adaptation is of tremendous significance for the recycling of inorganic nutrients in the world environment. We will be discussing four types of bacteria: Nitrosomonas, Nitrobacter, Bejotauta, and Methanogens. Nitrosomonas is a type of gram-negative bacteria which converts ammonia into nitrite. Nitrobacter is a type of gram-negative bacteria which converts nitrite into nitrate. Bejotauta is a type of fungi belonging to the order of Saprolenales. Methanogens are a type of anaerobic bacteria that break down biodegradable matter into methane gas. Bacteria can use light as a source of energy, but the carbon they need comes from organic sources. Two examples of this type of bacteria are Rhodospirillum and Rhodopseudomonas, which usually develop in areas with intense light. Palestric bacteria are a classic example of these bacteria. Bacteria can be divided into two groups: chemoheterotrophs and photoautotrophs. Chemoheterotrophs depend on organic compounds for both carbon and energy. These compounds are broken down through either respiration or fermentation, releasing the energy in the form of ATP, thus enabling the bacteria to carry out the necessary metabolic processes for their survival. An understanding of the two types of chemoheterotrophs is essential in the field of microbiology. Saprophytes are free-living microorganisms that obtain their organic nutrients from the waste of dead organisms, such as Bacillus sps, while other chemoheterotrophs obtain their organic nutrients from other sources. Being able to differentiate between these two types is useful when applying knowledge of microbiology in practical ways. Xanthomonas is a family of parasites that contains two very common representatives, namely Salmonella and E. coli, which can cause serious health issues if not taken care of. These parasites feed off of cells or tissues of a host and are one of the most widespread forms of bacteria. It is therefore necessary to be aware of their presence and act quickly. Adelovibrio bacteriovirus is a species of bacteria that lives as a parasite on some harmful bacteria. Believed to be responsible for the microbial purity of the water of the Ganges River, this is just one example of the many bacteria that belong to the two most important categories for biomedical research. With this in mind, understanding these microbes is essential in order to comprehend the implications of our research. Bacteria are an essential part of numerous ecosystems, with some of them belonging to the photoautotroph category. These bacteria use light energy to manufacture food from basic inorganic elements. Notable examples include Chromatium, Nitrosomonas, Nitrobacter, and Bejotoa. Bacteria have a fundamental role in the recycling of inorganic nutrients, particularly Chromatium, Rhodospirillum, Rhodopseudomonas and Nitrobacter. They are crucial for the cycle of life, decomposing materials and converting them to alternate forms. Without bacteria, life as we know it would be impossible. We focus on bacteria and their various nutritional sources in our last slide. We look at Xanthomonas, Bejotoa, Nitrobacter and Nitrosomonas, along with their respective nutrition that they derive from the cells or tissues of their host. We appreciate your time in learning about these various bacteria and their nutrition sources. Thank you.